So revision of chapter 12 and 13 real quick. See what is in the core of the chapter. So neuron and neuroglia. Neuron, normal cell, neuroglia, supportive cells. We have spinal nerves, we have cranial nerves, afferent and efferent. This is just the basics. We have receptor, we have effector, we have the somatic. So you need to know this. Somatic, what's the somatic and what's autonomic? And what are the two divisions of the autonomic? What's the difference? Sympathetic versus parasympathetic. Okay? Now the neuron. You need to differentiate between these types of neuron. Like, this is something that you have to know. This is the structure of the neuron. Okay? And we have different types of neuron that we will go to it. So unipolar, bipolar, multipolar. You need to know the difference between each one of those. Unipolar, bipolar, multipolar. Okay? You need to know what is it and the location. Is that clear? Where do we have the uni? Where do we have the bi? Where do we have the multi? The multi is almost everywhere. The uni and the bi are located in certain spots. You have to remember this.
So location?
Location of each one of those. Yes. Where is it located? Okay. The function of each of the organelles. Okay. What is nasal bodies, for example? Okay. Nasal bodies. It's Rafi R plus aggregation and so on. I'm not going to re-lecture again, but just understand the function of each part or organ. What's the dendritis? What is the axon? What's the axon hillock, for example? What's the axon hillock? This is the attachment of
The cell body
with the
axon.
axon. Yes. Dendritis, does it receive or send? Receive. So it moves toward the body, and the axon away from the body, right? So these are all parts of the neuron. So you need to understand this. Okay, you can study it, you can draw it by yourself. If you had this in the lab, it will help you because you need to know the different parts. Okay, this is very important. You have to know each one of those. What's the nucleus? And you're supposed to be familiar with this already from the organelles. I think it was chapter 3 or something, if you still remember. It's just the basics. I'm not talking about all the details, like mitochondria powerhouse, energy. Just simple word, energy. Rafi R, production of protein. Very simple, okay? Golgi apparatus, modifying and packaging. Just a simple. Neurotransmitter, very important. Neurotransmitter. What's a neurotransmitter? Something like acetylcholine. Exactly how it works. From the arrival of the action potential, to the entrance of calcium, to the release of neurotransmitter, to crossing the gap, to reacting with the receptor, to creating a depolarization by opening sodium channels and the postsynapse, to the degradation. How do we degrade acetylcholine? What's the name of the enzyme? Acetylcholine esterase. Yes. These are the different types, and this is the slide that I was talking about. Very important. You have to know which is anaxonic, bipolar, unipolar, and multipolar, and where is the location of each one of those. Okay? Okay, receptors are important. We need to know the receptors, interoceptors, exteroceptors, inside, outside, and position. Neuroganglia or neuroglia. Neuroglia. Neuroglia are not neurons. Each one of those has a function. Very important. And you can actually, to make it simple, you can get it from this. This slide is extremely important, okay? And I'm recording this, by the way, so you can go back to it. This slide is very important, meaning you have to memorize it. You have to memorize this slide, okay? What's myelinated versus unmyelinated? Important. Okay? Important. And the myelinated is white or gray? And unmyelinated? Okay. Which one is faster? Myelinated. Okay. Ganglia versus nuclei. Important. Both of them are masses of neuron cell bodies. But one of them is in the brain, in the central nervous system, I mean, and the other one is in the peripheral nervous system. Is that clear? So neuroglia versus nuclei. Okay. Wallerian degeneration is important. This is the basic way of regeneration. The steps, just understand the steps. Okay, any number here is important. Minus 70, minus 90, positive 30. Any number here is important. Like this. Minus 70 plus 66. This is equilibrium potential. Any number here is important. Enzymes, something like this is important. Sodium, potassium, ATPase. What does it do? And what's the ratio? We talked about this, remember? 3 and 2. Get 3 versus 2. 3, 2. Yeah, this is the sodium, potassium, ATPase. Okay, channels. The different types of channels is important. Is it a passive, which is leak, or is it an active? And what's the difference? Chemically versus voltage versus mechanically gated channels. So channels are important. Whether it's passive or active, whether it's chemically or voltage or mechanically gated. Okay? All or none principle. Okay, initial stimulus, all or none principle. We need to understand what does it mean. Absolute versus relative refractory period. You have to understand what's the difference. Very important graph. You have to know this graph. You have to know all the details about the graph. What is happening in each one of these points. Very important. Okay? Very important. You have to see this graph, understand it, translate what each segment means, and what each one of these number means, and what's the depolarization, repolarization. So this graph is extremely important. Okay? Propagation, we talked about this. Is it continuous myelinated or saltatory? Saltatory myelinated, I mean, or is it continuous? Which one is faster?
Saltatory.
The saltatory myelinated is faster, right? Which one is faster? The large diameter or the small diameter? Large diameter. Okay. The difference between graded potential and action potential is actually important. Just the basics, okay? The graded potential, the first potential that comes, just depolarization or hyperpolarization, versus the action potential, which is the actual depolarization. Okay? Axon diameter, we just talked about this. Large diameter versus small diameter. The differentiation between these three fibers, I think we have a table for this. Probably. Okay. Okay. I thought we have a table for this, but anyway, you have to differentiate between type A, B, C. If there is a table, we will see later on. If you have a table, just know the table. Type A, type B, and type C. And not only you understand the difference, which is it myelinated? Is it demyelinated? The diameter, is it large? Is it small? Not only that. These are the basics, okay? Myelinated or unmyelinated, diameter. Number 3, where? Okay? Where is it located? Where do we use it? Understand the difference between electrical and chemical synapse? Acetylcholine, we talked about this. The name of the synapse that come from the name of the neurotransmitter. So what's cholinergic versus adrenergic versus dopaminergic? So if you're using acetylcholine, it's called cholinergic. Adrenaline, adrenergic. Dopamine, dopaminergic. So the name of the synapse come from the name of the neurotransmitter. Okay? Synapse is important to understand, and we talked about this before in this revision. Starting from the action potential arriving at the synaptic knob, to the calcium entering, to the neurotransmitter leaving the vesicle, crossing the gap, going to the postsynaptic receptors, interacting with the receptors, opening the sodium channels if it is excitatory, or opening potassium if it is inhibitory. Okay? And then what's the fate of acetylcholine? We talked about this, right? This is very important. You have to know this. Synaptic delay. Okay. The name of these neurotransmitters is important, and the action is, so this is very important. Acetylcholine versus noradrenaline or norepinephrine versus dopamine versus serotonin versus GABA. In each one of those, this is very important, okay? The 5 neurotransmitters, very important. You need to memorize those. Is it clear? Acetylcholine versus norepinephrine. In each one of those, what is the name of the synapse? If it's adrenaline, adrenergic. If it is acetylcholine, cholinergic. If it is dopamine, dopaminergic, and so on. Number 2, is it excitatory or is it inhibitory? Number 3, where is it located? Very important, okay? Okay, opioids and neuropeptides are important. So you need to know at least the names. So now when I ask about the opioids, you need to tell me we have endorphin, we have enkephalin, we have endomorphin, we have dynorphin. Okay? So what are the neuropeptides versus opioids? You need to understand that. And you need to actually remember these names. The action is also important. Do you do a direct action or you mediate something in the middle? When you do the action of the neurotransmitter, like in this case, you're mediating the G protein and you're activating adenyl cyclase. This is the indirect. If it is direct, just go to the channel, open, close. That's it. The signal go to the channel open or close. Direct. Or it can be indirect using G protein and adenyl cyclase, which creates cyclic AMP. So this is indirect, G protein, adenyl cyclase to make cyclic AMP. This is the indirect way. The difference between the postsynaptic potential that I was talking about, we need to understand that. Is it EPSP or IPSP? What's the difference? Okay. The processing, important. Are we inhibiting or are we doing summation? And what are the types of summation, like temporal summation versus spatial summation? Temporal and spatial. So summation, this is how we do it. Facilitation. Okay, so what I focused on is not the only thing that you need to know. This is the core of the chapter. Okay, you focus more on this.